Functional Annotation and Molecular Modeling of Hypothetical Proteins, HPs, from P.aeruginosa plasmid PUM505, an in. Abstract structural and function annotation of P.aeruginosa plasmid PUM505 is essentially required to facilitate the understanding of mechanisms of pathogenesis and biochemical pathways important for selecting novel therapeutic target. In present study, randomly selected 12 hypothetical protein sequence of P.aeruginosa plasmid PUM505 has been annotated using various in-silico tools and databases to determine domain family, solubility of protein, ligand binding sites etc. 6 out of 12 proteins have been putatively annotated, in which 4 have been annotated with high confidence. Physio-chemical characterization revealed 2 proteins are stable. The three-dimensional structure of 2 important annotated proteins were modeled and their ligand binding sites were identified. Domains and families for 6 proteins have been found. The analysis revealed that these proteins have antitoxin activity, integrase enzyme activity, conjugal DNA transfer activity, etc. Structural prediction of these proteins and detection of binding sites from this study would indicate a potential target aiding docking studies for therapeutic designing against various diseases. Keywords: hypothetical protein, functional annotation, molecular modeling, docking. Introduction. Pseudomonas aeruginosa, a gram-negative bacterium, is well known for its environmental versatility. Diverse growing habitat includes soil, coastal, marine, plant and animal tissues. Con et al. 2007. P.aeruginosa is also well known for its multi-drug resistant and is a global threat towards many infections disease. P.aeruginosa is a major opportunistic pathogen in humans, causing serious complications caused by infections in patients particularly susceptible like people with immune system deficiencies, victims of skin burns, catheterized patients who suffer urinary tract infections and patients with respirators, causing nosocomial pneumonia. Lizik et al. 2000. It is the major cause of mortality in patients with cystic fibrosis colonizing the lungs. Williams et al. 2010. Role of plasmids in antibiotic resistance are well established. Plasmids are circular deoxyribonucleic acid molecules that exist in bacteria, usually independent of the chromosome. The study of plasmids is important to medical microbiology because plasmids can encode genes for antibiotic resistance or virulence factors. Wang et al. 1988. The PUM505 plasmid contains a genomic island with sequence similar to islands found in chromosomes of virulent P.aeruginosa clinical isolates. Plasmid PUM505 contains several genes that encode virulence factors, suggesting that the plasmid may contribute directly to bacterial virulence. Rodriguez et al. 2016. The bacterium's virulence depends on a large number of cell-associated and extracellular factors. The virulence factors play an important pathological role in the colonization, the survival of the bacteria and the invasion of tissues. Wang and Gui, 2013. Due to cost efficiency, throughput of genome sequencing has increased enormously, resulting thousands of bacterial genomes now available, and this number is increasing enormously day by day. Functional annotation of proteomes is a demanding problem. Roberts et al. 2004. A large fraction of proteins is still labeled as hypothetical protein unknown function, or with similar terms that imply that there is no functional indication for the ORF. Function annotation of putative uncharacterized HPs for their possible biological activity has emerged as an important focus for computational biology. Kumar et al. 2014, Lowenstein et al. 2009, Shabazz et al. 2013. The PUM505 sequence contained 138 complete coding regions, the majority of them encoded on the complementary DNA strand, 75%, with respect to the predicted origin of replication, or EV. Most of the identified genes, 46%, encode hypothetical proteins, HSPs. Proper structural and functional determination of this huge fraction, 46%, is very important to reveal complete understanding of virulence mechanism in P.aeruginosa. Therefore an improved functional annotation of its proteome is of particular urgency. In present study, 12 randomly selected HPs from P.aeruginosa plasmid PUM505 have been annotated with the help of various bioinformatics resources. Moreover, 2 important annotated HPs have been structurally modeled and characterized. Materials and methods. Sequence retrieval and physio-chemical characterization. Functional characterization. Functional annotation was performed using Conserved Domain Database, CDD, Marchler-Bauer et al. 2011, PFM, Bateman et al. 2005, and Eggnog, Powell et al. 2012, Cello, U et al. 2014, and Psort B, Gardayat al. 2003, were used for subcellular localization of proteins. Signal peptide, UT al. 2010, and secretome were used for non-classical secretory pathway in proteins. 3D structure modeling. Homology models of HPs were determined using Swiss model, Kieferit al. 2009, and Protein Homology Recognition Engine PHYRE2, Kelly and Sternberg 2009. The first server uses a library of known protein structures taken from the Structural Classification of Proteins, SCOP, database and the PDB, Kelly and Sternberg 2009. The top 10 scoring alignments were used to construct the three-dimensional structure of each HP. Validation of predicted model. The stereochemical quality of the modeled structure for HPs was validated with verification server PPSM, Laskowski et al. 1993, using PROC-HEC, Laskowski et al. 1996. PROC-HEC validates the stereochemical quality of a protein structure by analyzing the overall structure and residue-by-residue geometry of proteins. ERAT server, Lee et al. 2007, was used for structure validation. Active site prediction. Results and discussion. Sequence analysis and functional annotation. Physico-chemical studies of selected proteins revealed that 2 out of 12 proteins are stable. Most of the proteins are of acidic in nature. Supplementary Table S1. In pursuit of assigning putative function to hypothetical proteins, available sequences and functional information from various resources has been interrogated. 6 out of 12 selected hypothetical proteins have been putatively annotated using sequence/domain information from PFAM, functional information from EGG-NOG pathway, TABLE1. The presence of different domains in varying combinations in different proteins gives rise to diverse repertoire of proteins found in nature. Identifying the domains present in a protein can provide insights into the function of that protein. Protein YP_00492803.8.1 has been annotated as PIN3 family. Protein YP_00492803.1 Astrau family. YP_00492798.0.1 as Na37 family, and YP_00492798.9.1 as HNH endonuclease, Table 1. 4 proteins have been annotated with high confidant. PIN3 family protein plays a very important role and function as nuclease enzymes that cleave single-stranded RNA in a sequence-dependent manner. PIN domain contain 3 nearly invariant acidic residues. PIN domain proteins found in prokaryotes are the toxic components of toxin-antitoxin operons. These loci provide a control mechanism that helps free-living prokaryotes cope with nutritional stress. Arcuzet al. 2011. HNH proteins are involved in DNA homing, restriction, repair, or chromosome degradation. The HNH proteins are not only involved in homing but also carry other biological functions, such as DNA degradation, repair, and restriction. Walker et al. 2002. TRA-U is an essential protein to conjugal DNA transfer. Moore et al. 1990. All these annotated HPs play crucial role. Subcellular localization of protein is an important parameter for protein function in various cellular processes. Subcellular localization analysis revealed that 9 proteins are cytoplasmic, 1 outer membrane protein and 2 unknown. Table 2. Motifs are signatures of protein families and can be preferably used to define the protein function, particularly in enzyme where motifs are associated with the catalytic function. Bork and Kunin, 1996. 3D structure prediction and validation. Three-dimensional structures of 2 important annotated proteins have been done with the Swiss model and PHYRE2 server. Based on the results, the stereochemical evaluation of backbone and dihedral angles of the HSBs revealed that for model HP38, 94.6 and 5.4% residues were within the most favored regions, additionally allowed regions respectively, Fig 1b. No residue was found in generously allowed or disallowed regions. For model HP 80, 84.6, 12.8, 1.7 and 0.9% residues fall within the most favored regions, additionally allowed regions, generously allowed regions and disallowed regions respectively, Fig 2b. Overall 100% implies best stereochemical quality for both the model. Based on the stereochemical validation model, HP38 showed more robust model than HP80. Secondary structure prediction showed 5 helices, 2 strands, 7 beta turns for model HP03 while 7 helices, 4 strands and 7 beta turns for HP38, Fig 3ab. ARAT is a program for verifying protein structures determined by crystallography. ARAT server was also used for model quality estimation, which showed that overall quality factor was 77.11 and 81.32% for model HP38A and DHP80 respectively, which confirms as a good model. Fig. 1a-2a. Active sites in predicted models. Active sites on a protein is of fundamental importance for a range of applications including molecular docking, de novo drug design and structural identification and comparison of functional sites. During analysis, 7 and 4 binding pockets were identified for model HP38 and HP80, respectively, Fig 4. During analysis of NABPs in HP38 model, 2 largest positive patches were detected, viz. patch 1 GHE, SFN, LCS, FPK, PA, con patch 2, FPK, PA, LCS, con, GHE. For model HP38, 2 largest positive patches were detected, viz. PAC, GHE, SFN, LCS, FPK, PA, con patch 2, PA, FPK, LCS, SFN, GHE patch 3, PA, LCS, GHE. Results are shown in Table 3. This data of active binding site residues will give insight into identifying binding interactions and docking with specific ligands. Conclusions. Our primary sequence-based analysis led to the identification of 2 HPs as biologically significant, which might be involved as enzymes, antitoxins, conjugal DNA transferase, and oxidoreductase, etc. Furthermore, we successfully predicted the structure of 2 HPs to describe their functions at the molecular level. The outcome of the present study may facilitate better understanding of the mechanism of virulence, drug resistance, pathogenesis, adaptability to host, tolerance for host immune response and drug discovery for treatment of infections caused by P.aeruginosa. Acknowledgements. The authors gratefully acknowledge the financial assistance under Project Application of Microorganisms in Agriculture and Allied Sectors, AMAS, from Indian Council of Agricultural Research, ICAR, India. The authors further acknowledge the Integral University, Lucknow, for providing the necessary support for the completion of this study and allotting its manuscript communication ID. Conflict of interest. The authors declare that they have no conflict of interest.